So I wanted to do this video because I think a lot of people are going to think they are safe because they didn't get the COVID-19 shot. And people need to understand that this technology is going to be in everything. It says engineers design programmable RNA vaccines. Tests in mice show the vaccines work against Ebola, influenza, and a common parasite. This is from back in July 4th, 2016. So this is way before COVID-19 came out. It says MIT engineers have developed a new type of easily customizable vaccine that could be manufactured in 1 week, allowing it to be rapidly deployed in response to disease outbreaks. So I wonder why they were able to come out with a COVID-19 vaccine so fast. Now, people were saying all along that they had the vaccine. And, if this isn't proof of that, then I don't know what is. This is from 2016. So far, they have designed vaccines against Ebola, H1N1 influenza, and Toxoplasma gondii, a relative of the parasite that causes malaria, which were 100% effective in tests in mice. The vaccine consists of strands of genetic material known as messenger RNA, which can be designed to code for any viral bacteria or parasitic protein. These molecules are then packaged into a molecule that delivers the RNA into cells, which it is translated into proteins that provoke an immune response from the host. In addition to targeting infectious diseases, the researchers are using this approach to create cancer vaccines that would teach the immune system to recognize and destroy tumors. So, and you're going to hear more and more about cancer vaccines. It's already coming out, but it's going to get even more so. This nanoformulation approach allows us to make vaccines against new diseases in only 7 days, allowing the potential to deal with sudden outbreaks or more rapid modifications and improvements, says Daniel Anderson. Anderson is the senior author of a paper describing the new vaccine in the Proceedings of the National Academy of Sciences the week of July 4th. The project was led by Chahal, a postdoc at MIT's Whitehead Institute for Biomedical Research. Most traditional vaccines consist of an inactivated form of a virus or other pathogen. These vaccines usually take a long time to manufacture, and for some diseases they are too risky. Other vaccines consist of proteins normally produced by the microbe, but these don't always induce a strong immune response, requiring researchers to seek an adjuvant. RNA vaccines are appealing because they induce host cells to produce many copies of proteins they encode, which provokes a stronger immune reaction than if the proteins were given on their own. The idea of using messenger RNA molecules as vaccines has been around for about 30 years, but one of the major obstacles has been finding a safe and effective way to deliver them. Kahn decided to package RNA vaccines into a nanoparticle made from a branched molecule known as a dendrimer. One key advantage of this material is that the researchers can give it a temporary positive charge, which allows it to form close associations with RNA, which is negatively charged. Kahn can also control the size and pattern of the final structure by inducing the dendrimer RNA structure to fold over itself many times. Kahn generated spherical vaccine particles with a diameter of about 150 nanometers. That makes them of similar size as many viruses, enabling the particles to enter cells by exploiting the same surface proteins that viruses use for this purpose. By customizing the RNA sequences, the researchers can design vaccines that produce nearly all protein they want, or any protein they want. The RNA molecules also include instructions for amplification of the RNA so that the cell will produce even more of the protein. The vaccine is designed to be delivered by intramuscular injection, making it easy to administer. Once the particles get into cells, the RNA is translated into proteins that are released and stimulate the immune system. Significantly, the vaccines were able to stimulate both arms of the immune system, a T-cell response and an antibody response. In tested mice animals that receive a single dose of one of the vaccines shows no symptoms following exposure to the real pathogen, Ebola, H1N1 influenza, or Toxoplasma gondii. No matter what antigen we picked, we were able to drive the full antibody and T-cell responses. The researchers also believe that their vaccine would be safer than DNA vaccines, another alternative that scientists are pursuing because, unlike DNA, RNA cannot be integrated into the host genome and cause mutations. The option of rapidly creating a completely synthetic formulation that can be effective as a vaccine is an important addition to currently available vaccine strategies. The ability to rapidly design and manufacture these vaccines could be especially beneficial for fighting influenza because the most common flu vaccine manufacturing method, which requires the viruses to be grown inside chicken eggs, takes months. This means that when an unexpected flu strain appears, such as the 2009 pandemic causing H1N1 virus, there is no way to rapidly produce a vaccine against it. Typically, a vaccine becomes available long after the outbreak is over. We think we can become interventional over the course of a real outbreak. So, of course, that's what they did for the COVID-19. They say it's revolutionary because it could dramatically reduce the amount of time needed to respond to disease outbreak. This could not only be applicable to the bugs they talked about, but could also be applicable for something even more important, which is an unknown virus. In response to a pandemic, whether natural, accidental, or intentional, they could produce a vaccine in a week. It says, we are all excited about the potential for the new approach to provide a new way of vaccine delivery. The research was funded by the Department of Defense Office of Congressionally Directed Medical Research Joint Warfighter Medical Research Program, Medivector Incorporated, the Reagan Institute of MGH, MIT, and Harvard, and Defense Threat Reduction Agency Joint Science and Technology Office Program in Vaccines and Pretreatments. So, these people that were saying that they already had the vaccine out, I believe they're absolutely 100% correct. But what I do find interesting is people don't really tend to talk about anything outside of COVID-19. And it seems that if you avoided the vaccine, people have the idea that now they're safe. But if they think that they can avoid the COVID-19 vaccine and go out and get other injections to prevent them from getting cancers and other things, they're wrong, because it's going to be the same design. It's going to be the same design and everything, and it wouldn't surprise me if they start putting it in pills as well. As always, put your faith and trust in Jesus, repent of your sins, and have a blessed day.